learn about the meiosis phase, first I'll start with the interphase. Okay, suppose this is a cell. Okay, now it has what? It has a pair of chromosomes which are homologous in nature. These are homologous chromosome. What I told you about homologous chromosome? The chromosome which are same in shape and size and have the same centromere position, and also they have same length and the staining pattern. They are what known as homologous chromosomes. Okay, now consider it is a diploid cell. It is a parent diploid cell. Okay, which has pair of homologous chromosome. Now what happened is duplication happens. Here the chromosome get divided. We can say duplicated. Now these are called duplicated pair of homologous chromosome. And you know, when the chromosome get duplicated, it had what called sister chromatids. Sister chromatid get formed. Okay, now it's still an adult cell. It is diploid in nature right now also, with what? Duplicated chromosomes. Till this phase, it is termed as interphase. Okay, now let's step further. What happened is, now the cell divide. This cell will divide into two cells. Okay, what happened now? What you can see, you see that the chromosomes separated, the homologous chromosome gets separated. What do you think? What happened? The division of cell here done here, and the cell divided into two, and the chromosomes separated. This phase we can term as is meiosis I. Why? Because when the adult divides, diploid becomes haploid, 2n 2n. Okay, still we got the duplicated chromosomes, but in haploid cells. Okay, the main event was homologous chromosomes. I'll just write it as HC get separated into two different cells. Now further division took place. What happened here is, these are called sister chromatids. Okay, what do you think? What happened? What do you think? What happened here? I gave you a hint. See this. See this. Yeah, you guessed it right. Sister chromatids. I'm writing it SC. Get separated into what? Four cells. These are still what? Haploid. But now they are not duplicated. They are unduplicated chromosomes. We have four haploid cells with unduplicated chromosome now. This event, we can mark this phase as meiosis II. a lot of people will ask, why in the first phase, like in the from interphase to meiosis 1, the 2n becomes n. Okay, but in the meiosis 2, why the n doesn't get half? Why? It still remain the n. To answer this, what happened is, in the first cell, we know there are two chromosomes. Okay, the cell get half. Okay, it divide into half, so the each cell got its chromosome. Okay, its duplicated homologous chromosome. We term this as what? Meiosis 1. Here you know it is a parent cell. In the parent cell divide is divided into n and n, to haploid daughter cells. Okay, but now here we can see the division again happened, but this time the chromosome number, the chromosome get divided. So four new chromosomes, or four, we can say that nearly identical, we cannot say identical, four sister chromatids, here sister chromatids, these one are sister chromatids, these divided further, so that's why the number gets same. If the cell divided again, okay, then the number become half, but here the cell doesn't divide. So we call meiosis 1 as what? Reduction division. Why? As 2n reduces from n to n, reduction division. But in meiosis 2 we call it equational division, as n remains the same. They remain the haploids. So what is the final result? Okay, the final result we can see is four haploid daughter cells, each with what? Each with half of the chromosome of the parent cell. Each with, you can see it now here. These are like, this is one parent. Okay, this get half in the meiosis 1 and half again in the meiosis 2. The key thing to note here is only in meiosis 1 and meiosis 2. Meiosis 1, what happened mainly? Homologous chromosome gets separated, and also the chromosome number reduces from diploid to haploid. But in the 2, only the sister chromatid gets separated. Sister chromatid gets separated. So that's why chromosome number is same. Now in the four haploid cells, each contain one chromosome. Like, difference between sister chromatid and the homologous chromosome is, sister chromatid are what? Two identical copies of one chromosome. What are homologous chromosome? It is like one chromosome from mother plus one from the father side. They are joined at centromere. But in the case of homologous chromosome, they are similar in shape and size. They carry different alleles. I'll talk this about in next lecture, or we can say, okay, I'll just tell you now. Alleles are, the version of the same gene. They are not physically attached except during the meiosis. Now, sister chromatids. They are formed by what? After DNA reapplication. They are held together by sister chromatid cohesion, I told you earlier. So together, sister chromatid is one duplicated chromosome. Now let's talk about the alleles. They are like they're called as different version of the same gene. Different version of the same gene. Why? Because like it is located in the same locus. It is located on the locus on homologous chromosomes. For example, these are two homologous chromosomes. At this point, in this chromosome and this point, same in other chromosome, the eye color gene is attached at the same point. Okay, this allele gives the brown color. It is formed at the same locus. We term this place as locus. So that's why they are called allele. But you know, is it the eye color gene? It is the eye color gene. It is the eye color gene. But one of the gene here, like for example, this gene gives the brown color. This gene can give the what? Blue color. But they are attached at the same line. So that's why this is called an allele. Now let's talk about the key difference between like the mitosis and meiosis. Meiosis and mitosis. We learned that far till now. Okay, in meiosis we learned that okay the chromosome divides. The chromosome duplication, right, at CD. Okay, it divides what? In ones. In the meiosis, in mitosis also we know that it divides only once. Cell division. How many times in meiosis? Two times the cell divides. First in meiosis 1, secondly in meiosis 2. Mitosis still once. We can say 1. Okay. Next thing, chromosome number. Chromosome number. Write it as CN. In mitosis, it is the same as parent, but in meiosis, half of the parent. Okay. Daughter cells. In mitosis, there are two daughter cells. Cell formed into two, divided into two daughter cells with identical nature, but in meiosis, there are four. After in meiosis 1 and meiosis 2. Lastly, what happened? Genetically, genetic makeup we can say. In mitosis, it is nearly identical, same nature. Okay, but meiosis it is genetically different. The babies, we can say the offspring are different. The important point we can note in from here only, because like the chromosomes get duplicated only once. Okay, meiosis involves divisions, two divisions. Mitosis involves one. That's it for today. Then meet you in the next video. We'll discuss the interphase, the anaphase, the metaphase, telophase, cytokinesis, and we'll discuss like how the sister chromatids get separated further. Okay, and the crossing over, the synapses, synaptonemal complex, and that's that's it. Bye bye for today.